Hello everyone. Let's look at the next stage of residual management. In the first step or the first stage, we looked at thickening, right? We wanted to increase the concentration of the solids either by gravity thickener or by dissolved air flotation. So, the next aspect is, now that I have relatively more concentrated solids in my sludge, I want to decrease the biodegradability of this particular sludge. As in, if I leave it out there, again it is going to stink because of anaerobic conditions. I do not want that to cause nuisance. It can attract flies and other kinds of what do we say, insects and such too, right, or other insects too. So, let us look at the different ways we are going to achieve stabilization by. So, different methods. One is anaerobic. Firstly, why anaerobic? And again note that this is widely used. Sludge, we have, especially with the organic sludge here, we have a lot of organic concentration. So, if you want to do it by aerobic, right, you need to provide oxygen. If you are doing it within a controlled what do we say, system, yes, that also can be done, but again you are giving or needing to put in energy, right. So, but with anaerobic digestion though, depending upon the byproducts or the extent of optimization you are able to achieve, you can get products such as methane, which can then be used for energy generation or for cooking or such. But again it depends upon the calorific value of your particular gas, let us say. So, that is one aspect to note, anaerobic digestion, let us see. Other one is aerobic digestion, which we will briefly look at later. Autothermal aerobic digestion and composting, which is done in open land, you would have seen this, and lime alkaline digestion, this also we will look at later. For this session we are going to look at anaerobic digestion, let us see, right. So, let us move on. There are different stages here, right, and note that my sludge has all these relatively complex organic matter. So, complex stuff, well, I can't eat, I can't digest, right, or remarkably complex products. Typically that is why I am going to cook it or such. Well, it is not the same analogy, but even then, even similar or similarly for the microbes too, remarkably complex organics, they can't degrade or use them well enough, right. So, first they have to be, what do we say, changed into forms that are relatively more soluble and also relatively easily used or usable by the relevant microbes as the substrate, let us say. So, what do we have? Major what do we say, compounds or types of compounds of interest? We have lipids, polysaccharides, we have proteins and nucleic acids, let us say, right. So, the first stage is hydrolysis. Typically this is brought about by the enzymes of the microbes which are prevalent in this particular system. So, that is one aspect to keep in mind, hydrolysis, right. And then you are going to have fatty acids from lipids, monosaccharides, earlier it was poly and now monosaccharides, protein to amino acids, right, nucleic acids to purines, pyrimidines and simple aromatic compounds. So, here we have hydrolysis, and if I am trying to, let us say, use kitchen waste, right, kitchen waste, as in now the sludge, not sludge, pardon me, kitchen waste, we are dumping it out there, that is leading to huge landfills because it is going up with the inert waste and leading to huge landfills, right, and there it is going to be an issue because of the kind of organic and inorganic content there, I can't really treat that well. So, now if I can separate it out well and I have only the organic waste after segregation, then I can again have the anaerobic process too, right. I am just talking about another aspect which is relevant here, at least in the Indian context. So, that organic waste too I can have or let it undergo anaerobic digestion and get energy from it, but their hydrolysis will be pretty much slow because of the type of waste. So, typically people grind it with water or the sludge and create a slurry, right. So, hydrolysis is one aspect and that can be typically slow depending upon the type of compounds that you have at your inlet, let us say. So, now you have relatively simpler products here, let us say, not products, forms of organics, and then here we have two stages. One is acidogenesis, acid, and the other one will be acetogenesis, right. One is acidogenesis, right, acid, and the other one is acetogenesis, right. So, that is something to keep in mind. We will look at what it is that we are going to look at or see here. So, as I mentioned here, it is going to be two aspects in conjunction, and typically we are looking at fermentation, let us see. Note that here there is no oxygen involved here. In a redox process, you are going to have transfer of electrons. In the aerobic activated sludge process, oxygen was the electron acceptor. Here, if it is fermentation, the same compound will be lead to a relatively more oxidized and relatively more reduced compound. Or in the case of methanogenesis, you can have hydrogen as the electron donor, carbon dioxide as the electron acceptor and such, let us see. So, that is something to note, we will come back to that later. So, what is it that we have formed during the fermentation step? You have relatively complex fatty acids like propionate, butyrate, lactate and such, let us see. And then they are going to lead to formation of acetic acid, I guess, right. So, that is one aspect to note, that is why acetogenesis, if I am not wrong, but we will come back to that. And after that you are going to have methanogenesis, right, as in methane is going to be formed in this step from methanogenic substances, right, and hydrogen is essential, we will see why, and carbon dioxide too is going to be formed in one of the products, but we will look at it. So, 4 major stages, one is hydrolysis, acetogenesis or acidogenesis and acetogenesis, both of which are under fermentation, and then methanogenesis, which is leading to the formation of methane. Let us move on. So, first I have relatively more complex, what do we say, organics here, and this is by COD, this percentage is by the total COD, let us see. So, initially I have 100%. So, after hydrolysis I am going to have relatively simpler, what do we say, organic compounds. Again you can see obviously the COD is still there, right. And then what is it that is being formed? In the acidogenesis, you are going to have intermediate volatile fatty acids being formed, right. And in the acetogenesis though, you are going to have acetic acid being formed, either directly some of it from these compounds after hydrolysis, or from the volatile fatty acids or the intermediate volatile fatty acids that were formed. So, now you see there are two stages, one is acidogenesis, when you have the intermediate volatile fatty acids or some of it directly going to acetic acid, and the other one is acetogenesis, I guess, right, where as I mentioned you are going to have formation of acetic acid and also the transformation of all these intermediate volatile fatty acids into acetic acid. And note that in this particular process or during this process you are going to have hydrogen being formed, right. So, that is something to note. Why is that? Later this hydrogen is going to be required for the formation of methane, right. So, that is something to keep in mind. This methane production requires acetic acid, hydrogen, and if there is CO2, that too, I guess, right. So, one aspect to note is that if the hydrogen concentration is too high here, too high, or if it accumulates, right, there are 3 or 4 steps, one is here, and if I can combine all this as one step, and this is the third step, let us see. If sometimes this is too slow and this is too fast, what is going to happen? The hydrogen concentration is going to increase. If the hydrogen concentration is going to increase too much, let us say it is A plus B going to products C plus D, and one of this is hydrogen. Initially for this reaction to go through, right, if the delta G is negative, that is when the microbes will try to facilitate this particular reaction. But if one of the products has accumulated because the hydrogen or D has not been used later, D plus what do we say, X goes to Y, let us say, right, this is the methane, let us say, okay, because D has been not been consumed, let us say, and D is accumulating. So, what is going to happen? Delta G will not be maybe favorable, it might even turn to be positive. So, then the whole step will be, or the whole process can come crashing down. So, that is something to keep in mind, as in there is a fine balance between the hydrogen that is being produced here and the hydrogen that is being used up by the methanogens or during methanogenesis. So, that is something to keep in mind, right, and from here you are going to end up having methane, right. So, let us move forward. So, I guess this is the one from the MIT aspect, but I just wanted to mention it because it is a relatively simpler figure. Complex organics to higher organics, right, hydrolysis and or fermentation, here they clubbed it, but note that there are two aspects, and then acetogenesis and dehydrogenation, as in you are going to sometimes have formation of the hydrogen too, right, or use up of the hydrogen too. So, again key note is that we have acetic acid and also hydrogen. And another aspect to note is that acetic acid CH3COOH, right, it has a pK of 4.76, and note that this process is sensitive, one process is sensitive to pH of around 6.5, the methanogenesis, the other one to around 5.5, let us see. So, if the pH falls considerably, and especially when you have sulphide, right, and the pK of this around 6.7 or I think 7, let us say, right. We know that hydrogen sulphide something like this. Right. This pKa is around 6 or 7 if I am not wrong. So, if the pH falls to around 6.5 or such, most of it will be present as H2S, only some as HS minus, the other as S2 minus, and this H2S is toxic to the microbes. So, here similar to us, the microbes are very picky, right, and that is why the microorganisms, especially the ones that lead to methanogenesis, the methanogens are called the prima donna, right. You need to really take great care of them. Any changes to their particular what do we say environment, and they will start what do we say nagging at you, right. So, these are the prima donnas, I guess, right, of this anaerobic digestion process, where the pH and also the toxicity of some of the relevant products which is very low in concentration or which are very low in concentration too is important. So, let us move forth and see what else we have. So, hydrolysis, it is lysis, right, we are breaking something, lysis. Complex organics are broken down to simpler compounds, again which are more soluble, and then they can be consumed by the various bacteria, let us see. And acidogenesis, what do we have? These fatty acids, FATTY, fatty acids and alcohols are oxidized, and amino acids and carbohydrates are fermented to form volatile fatty acids and hydrogen, right, volatile fatty acids and hydrogen, right. For example, I guess glucose 2 acetic acid, this is one particular example, but this is directly with respect to acetic acid formation. We could have chosen a better example too. Let us look at that, we will come back to that. And then the other one is how is this done? We have acidogenic bacteria. They start quickly and can produce volatile phosphoric acids faster than methanogenic bacteria. So, this was the key too that I mentioned earlier. So, in this process, if hydrogen accumulates too much and is not removed by these methanogenic bacteria, which can grow slowly or take time to grow, then your system is going to fail, right, or lead to what do we say, it is not going to be beneficial to the microbes to take the reaction forward. Why? The delta G is now maybe not favorable anymore, that is something to keep in mind. So, one aspect to note is that, as I mentioned earlier, fermentative and acidogenic bacteria are inhibited, but the inhibition occurs at much lower pH than the pH at which methanogenic bacteria are inhibited, which is at around 6.5. But let me not digress. The acidogenic bacteria which take care of acidogenesis are inhibited at pH 5 or less than 5. As can be seen, they are robust and they can keep producing volatile fatty acids even when methanogens are not really what do we say thriving, let us say, right. But the key to successful digestion, as I mentioned, is from this point of view keeping acidogens and methanogens in balance. So, you will have to monitor the pH to keep track of the system, right. And acetogenesis, conversion of these intermediate fatty acids or the complex fatty acids to acetic acid, that is why it is acetogenesis, right. So, let us look at some of the relevant steps. So, this maybe we can look at it. So, earlier if I had a breakdown of a complex compound during acidogenesis into propionic acid, let us say, do I have that here, or what did I have here, or did I have it directly to acetic acid here? Okay, I had it directly to acetic acid, that is fine. So, some of these volatile fatty acids or intermediate volatile fatty acids can be propionic acid, and that now we are going to undergo acetogenesis, and you are going to have formation of acetic acid here, and as you can see, hydrogen and carbon dioxide are given out too. Note that hydrogen is starting to accumulate, let us say. And then methanogenesis, this acetic acid which is formed here is going to be converted to methane and CO2, right. So, let us see what happens. So, there are two different kinds of microbes in play. One is the one that leads to formation of CH4 and CO2 from acetic acid, and the other one where CO2 in the presence of hydrogen leads to formation of CH4 and water, let us see. So, here electron acceptor is CO2, here electron donor is H2. So, obviously, you see that H2 where is it coming from? This H2 is coming from here, and if because the methanogens are not thriving and this does not occur, this will also lead to accumulation of this hydrogen here in this system, and then this reaction which I want to go through is not going to be favorable for the microbes, right. So, that is one thing. As I mentioned, delta G should be negative, and if too much hydrogen or one of the products accumulate, that might not be the case, right. So, let us move on. And methanogenic bacteria, as I mentioned, are the prima donnas, and why do we need them? These reactions, note, are ubiquitous in nature. For example, if I throw out any organic matter, you are going to see methanogenesis, but they are not very much at high concentrations, right. They are at low concentrations. Though they are ubiquitous, they are present everywhere, but they are present at low concentrations, right. Generally, in an anaerobic digester, you have to seed it. So, you have to get that seed from somewhere else and then put it up here, right, anaerobic digesters. So, that is something to keep in mind. As I mentioned earlier, they are very sensitive to pH, right. Typically, neutral pH is 7, and if pH is less than 6.5, you are going to have considerable effect. So, that is why alkalinity has to be looked at. Why? Because if the pH is falling down due to, let us say, maybe the formation of acetic acid or any other particular compounds, right. Again, your acetic acid pK is 4.76, keep that in mind, right. So, if this is acetic acid HAAC, I am using that. Acetic acid is CH3 COOH, right, acetate and H. So, HAAC, and the deprotonated form is AC minus, let us see, right, AC minus. So, as I said, this is 4.76. So, at most of the pH, right, or pH 7 or 6, you see that AC minus is going to predominate, or HAC will give out its H plus. So, pH will fall down. So, if I do not have enough alkalinity in my solution to neutralize this H plus, that is going to be, what do we say, formed in the system, then the pH will fall down. And if the pH falls down, what is going to happen? The prima donnas, the methanogens, are going to be affected, and then the whole system is going to be affected. Why? The hydrogen is now not being consumed. If the hydrogen is not being consumed, the bacteria that lead to acetogenesis or are involved in acidogenesis, right, they will not find that particular reaction favorable anymore, let us say, right. So, that is something to keep in mind. It is a knock on effect. So, let us see what else we have. As I mentioned, prima donnas, and they are very sensitive to temperature and pH too, right. So, let us just look at the generalized reaction. So, you have the organic matter, carbohydrates, proteins and such, substrate and source of carbon. And what is the typical electron acceptor, right? It can be sulfates, nitrates, carbonates or such. And anaerobes, what are they going to use it for? For formation of new cells, energy for the cells, right? They have to thrive. One is reproduction, one is the energy for the cells being functioned, and in that process you are going to have CH4, CO2 and some other end products like H2S, N2 and H2. Why H2S? Obviously, if sulfate is there and you have anaerobic conditions prevailing, sulfate can act as a electron acceptor, right, and if it is acting as an electron acceptor, it is going to be reduced and you are going to have formation of hydrogen sulfide. As I mentioned, this hydrogen sulfide can lead to toxicity for the methanogens and affect the whole system. So, this is one aspect to understand, let us say, right. So, let us just simplify it one last time, I guess. Breakdown pathways in the hydrolysis step: carbohydrates going to simple sugar, alcohols, aldehydes and organic acids. So, proteins, you are going to have amino acids and then again obviously source of nitrogen, fats and oils, organic acids, let us say, right. So, let us see. As I mentioned, alkalinity of the sludge is very much important. Why is that? Acetic acid, right. So, as you can see, organic acids or even acetic acid, right, they are going to lead to soaring of your anaerobic digester. There is a term called soaring. So, that is one aspect to keep in mind, right. So, here you have, what do we say, HCO3 minus, the one that is going to be prevalent at pH 7 or near around pH 7, right, the inorganic carbon that can take up H plus, right, and this way the pH is not affected, let us say, right, or here I guess they have it presented in a different form, okay, CO2 instead of H2CO3. So, I guess with that I will end today's session. So, one aspect to note is that anaerobic digester, you have to be, what do we say, catering to the needs of the system primarily with respect to pH, hydrogen buildup and the methanogens which are very sensitive to the temperature, right. So, these are the aspects that have to be taken care of. So, in the next couple of sessions we will look at the other aspects after or the different kinds of stabilization, and then more or less finish this particular course. Thank you.